This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 305, recorded on February 22nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
I don't have my phone. I can't even tell you what the weather is. I have no idea, but it's pretty nice out.
It's pretty nice here as well. We have about 55 degrees, moving into that sweet spot of almost 20 degrees C, and it's sunny, and everybody seems to be happy.
Yeah, just wait. It'll change.
Yes.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, where it has been a rather freaky, balmy, like 50, 60 degrees the last couple days in February.
It's not normal, you're saying?
It's not normal.
Okay. And from St. Louis, Missouri, Petra, Levin.
We have a gray but 65-degree day, so
my gosh.
we're warmer than South Carolina. It is weird. It does happen to us sometimes in February, but it seems to be happening more and more. So my flowers are very confused.
Yeah, they bloom and then it freezes and they
die.
They die.
I think the daffodils are okay. They're pretty tough. But
all right. For your listening pleasure today and your education in the world of microbiology, Michelle's going to tell us about a cholera outbreak.
Yeah, for the last couple of years, there's just been a terrible explosion of cholera cases in Africa, in particular Central and Southern Africa. And this will include the countries of Zimbabwe and Zambia. And more than 85% of the world's deaths due to cholera have been happening in this region. So I want to first thank the two reporters from the New York Times, John Elligan from Johannesburg and Jeffrey Moa, who has been reporting from Zimbabwe in an article published February 13th in the New York Times about this. And then that led me to learn more about it. So what they've described is that there have been a number of severe storms, heavy rains, and flooding due to climate change, plus malnutrition, and then people being displaced by natural disasters or armed conflicts puts them at risk, because we're in regions of the world where sanitation is poor, where the water used for drinking, for food, for washing is not separate from our wastewater. You're at risk of cholera because this is passed by the fecal-oral route, which means people shed it in their stool, and then if the stool contaminates drinking or water used for cooking or cleaning, people are at risk. So this has been getting a lot of attention. The Africa CDC held a summit of the Southern Africa Development Community in February, and they reported that in a 13-month period ending February 2024, there were more than 250,000 cases and more than 4,000 deaths due to cholera in this region. 19 African countries were affected, more than 70% of the cases in Southern Africa. So they've made a number of recommendations, but before I get there, let's back up and talk a bit about what is this bug cholera. So it is a gram-negative environmental bacterium that, as I mentioned, spreads by the fecal-oral route. The bug is Vibrio cholera. There are more than 200 serotypes that circulate in the environment, but interestingly, only 2 of those serotypes cause outbreaks, and they're called the O1 and the O139. And the disease is basically a diffuse, watery diarrhea. People shed liters of fluid in a short period, due primarily to a toxin that the Vibrio make, best known as cholera toxin. So when a person, especially a child, sheds that amount of fluid in a short period, they're at risk of dehydration and shock, and it can be fatal. So the therapy is to rehydrate as soon as possible with water that contains salts and sugars. And that unfortunately has been in short supply. Now, here in the United States, we used to have cholera back in the 1800s, but since we built out our municipal water and sewage treatment systems, the cases in the U.S. are primarily people who've traveled to regions where cholera is endemic. It is rarely contracted in this country on seafood that's been imported and that is contaminated, because this bug can attach onto the shells of small and large seafood. But the FDA now does test both imported and domestic shellfish for contamination with Vibrio cholera. So this is a preventable disease. Here in the U.S., we're able to prevent most of the cases. And yet, globally, each year, there are between 1 and 4 million cases of cholera still, and between 20,000 and 140,000 deaths. We do have vaccines. In the U.S., there is a live oral vaccine that's been approved by the FDA. It is against the serogroup 01 type, but it's only for people aged 2 to 64 who have plans to travel to a region that has endemic cholera. And then globally, there are wholesale inactivated oral vaccines that have been approved by the WHO. But we learned from the recent summits on this topic that they've actually been in short supply. So they estimated that they needed 60 million doses to counteract this ongoing outbreak, but only 37 million are currently being produced. So right now, Doctors Without Borders and the WHO are bringing healthcare workers in to provide this hydration therapy to replace the fluids that are shed and the salts and sugars. But clearly, this is not enough. And so the Africa CDC had, as I mentioned, brought together this summit of the Southern Africa Development Community, and they've made a number of recommendations. Number one, we need to improve the public health surveillance so that we have early detection and warning systems so that we know that when cholera is present and spreading. A vaccination campaign for the vulnerable people, and also, of course, expanded production of this vaccine so that all the people that need it are able to get it. And then importantly, as was demonstrated by the history in the U.S., we need to develop resilient water, sanitation, and hygiene infrastructure. And the community abbreviates this as the WASH policy. So water, sanitation, and hygiene infrastructure, because that's a preventative measure. And then also increase access to the oral hydration therapy. So when there are outbreaks, there is treatment available. Now, there was recently an article also in Current Opinion in Pediatrics published in January of 2024 from investigators at the University of Nigeria. They point out that 14 million children currently are at risk of cholera, especially those under the age of 5. And they too made a series of recommendations which are that parallel those by the Africa CDC. So this is really a public health emergency, and it calls for a number of public health measures to be instituted, obviously funding to support these countries, and increased production of the vaccine, which can reduce the death rate.
So you say the can reduce. Do you have any idea how good the vaccine is?
The papers and the CDC webpage also has data on that, but I don't have those numbers on the top of my head.
But I think the best solution is to make certain that in these areas where there is active cholera, there's a wide availability of the oral rehydration salts.
Right.
They're extremely inexpensive. The standard recipe is 3.5 grams of sodium chloride, simple table salt. Potassium chloride, 1.5 grams, trisodium citrate, which is nothing more than citric acid, which is found in every popular soft drink that's consumed globally, 2.9 grams, and then glucose, 20 grams of glucose to facilitate the uptake of the electrolytes. This can reduce mortality to less than 1%. I have a colleague in my institution who is from Malawi, and this is where cholera is actually raging. They have the largest number of cases in Africa. And she and I were team teaching evidence-based dentistry to the dental students on Tuesday, and I was asking her if she has heard from any of her family members about the cholera outbreak, and she says it's horrible. Malawi's had 58,000 cases in 2023, and over 1,700 people have died.
Yeah.
And I asked her about water and sanitation. She says, well, we have that, but the problem is once it starts, it's so hard to stop. And, you know, the article in the New York Times is interesting. If you look at the photograph that's in the web version, you can actually witness what a cholera cot is. It's literally a cot with a hole in the middle that the patient sits over, and they literally drain 20 to 30 liters of fluid per day. 20 to 30 liters.
Explosive. Those of you who've had colonoscopy will have some familiarity with the feeling.
Yes.
I mean, it's terrible also because, I mean, the problem is, like you mentioned, that once you get it into the water supply, it's very difficult to get it out.
Yes.
And the amplification in each human is huge. So in those liters that people are shedding are lots of Vibrio that are ready to move on. And Michael, you make a good point that the hydration therapy is straightforward, but we have to keep in mind these are a lot of rural areas that are getting their drinking water from natural lakes and streams. And when there's heavy storms, the pit toilets that are common in those areas can then overflow and contaminate the lakes and streams that are really their essential sources of drinking water. So we are spoiled that we can go into any, 7-Eleven or whatever and
get a bottle,
gas station and grab sterile water, but it's not everywhere, and we need to do a better job of supporting infrastructure.
It's going to be hard to do that because if you look at the photos in the New York Times, I mean, it's just
it's devastating.
Everywhere. The vaccine is really going to be what's going to be
savior
effective now, because to fix that water system is going to be really a big job.
Yeah.
But the vaccine is not lifelong immunity. It's only effective for a limited amount of time. We don't have good data on the new ones of how long they are. They have an estimate, but as you say often on TWiM, we need the real data to see how long the vaccine is actually effective, because this Vibrio will change its O antigen in order to adapt. The other thing, what's interesting,
so Michael, you're saying that it undergoes antigenic variation, and that makes the vaccine not work.
That's why we have so many.
But really, only 2 of the serotypes are classically cause outbreaks: the O1 and the O139.
Because they are carrying the prophage that has
the toxin,
the toxin gene. The other interesting piece of the biology is that often these Vibrio live inside amoeba. And Rita Caldwell, probably now over 30 years ago, demonstrated that you can actually filter out Vibrio contaminated amoeba just by passing them through sari cloth, a crude filter. And that would often reduce the dose of Vibrio that the individual was ingesting to a level where they wouldn't contract the disease. But as Michele pointed out, it's really about groundwater and where you get your groundwater and whether or not your groundwater has been contaminated, because often people are still infectious 2 to 3 days even after they recover. And as we learned in Haiti, individuals can become carriers of cholera, so they can move it around the neighborhood, so to speak, depending upon where they happen to be going to the bathroom and relieving themselves. And that's why it's so important to follow this wash strategy that the WHO has developed.
Sobering.
There's also a parasite infection that is removed by the sari. I can't remember what it is, but for people that collect water, sometimes when they walk to the water, it's many miles, right? And they get blisters on their feet, and they put their feet in the water, and the blisters actually are parasitic blisters, and the parasites go in the water and contaminate it.
Guinea worm.
Guinea worm, right.
Yeah.
And so just passing it through a sari will fix that also.
Yes. Guinea worm is, the Carter Center has been trying to eradicate guinea worm, which is much more straightforward to eradicate than cholera. So.
Yeah. All right.
We're not there yet, but it's gotten much better.
Well, thank you, Michele.
Yes, you're welcome.
Now on to a paper from Petra.
Okay, so today we have a paper by Wu et al. It's a long list of authors, but Meng Wu is the first author. Wen Zhang, Xinyang Song, Bin Bao, Yuan Yuang, Deepshika Ramanan, Daping Yang, Rui Lu, John Macbeth, Eliza Doe, Morrison Andrade, Tiandi Yang, Yang Su Cho, Francesca Gazzaniga, Marit E. Ives, Daniela Coronado, Charlotte Thompson, Sayu Hang, Isaac Chiu, Jeffrey Moffat, Ansel Tsao, John Michelanos, Christopher Benoit, and Dennis Casper, entitled Gut Complement Induced by the Microbiota Combats Pathogens and Spares Commensals.
And if I could just interject, Meng Wu and Wen Zhang are actually co-first authors.
Yes.
And we'll learn more about them as we go along.
Right. And they are both Meng Wu is at in the microbiology department at Washington University Med School. So right on the other side of the park, experiencing the same weather we have today here. This paper has actually, as Michael pointed out, a really nice graphical abstract for those of you who can't access the paper or just want the cliff notes. It's a very nice graphical abstract that explains sort of the point of the paper and their results. But the big question of this paper is is there complement in the gut to modulate the composition of the microbiota? And in addition, how does complement, if it is in the gut, differentiate between pathogens and commensals that should be part of your gut microbiome?
The friend or foe question.
The friend or foe question. And I think the most important things here are, first of all, that they actually do find complement, and they do find actually that complement, at least the sort of stripped down version of complement that they find in the gut, does actually kind of help prevent pathogens from gaining a foothold, which is very cool. But before I get into the details of the paper, I think it's important to understand what complement is. So complement are proteins that were originally found in sera. So if you have blood and the red blood cells sink to the bottom and you have this straw-colored liquid on top, that's sera, classically. And these proteins are able to attack microbes independent of cells. There's two pathways. The alternative pathway, which is part of the innate immune system, so it's not specific or very specific, and the classical pathway, which involves antibodies. And the alternative pathway, which we just taught in the intro microbiology course, which I co-teach to WashU undergraduates, I always think it's one of the coolest parts of the immune system because you have essentially proteins that recognize pathogen-associated molecular patterns, or PAMPs, and these are evolutionarily conserved molecules on the cell surface of gram-negative and gram-positive bacteria, and they recognize those PAMPs, and they do three things once they recognize them. Again, these are just proteins. They don't require any cells. They're just found in the cell-free sera. So the first thing the complement proteins do is one protein that is actually the focus of this paper called C3. It actually starts to coat the pathogens, and I think it can do this to some viruses, envelope viruses, and it basically makes them look more tempting to phagocytic cells, particularly neutrophils, macrophages. I think of it as like putting a little salt on the pathogen, so the phagocytic cell is like, oh, that looks tasty, and we'll eat them. So that's one function. A second function is that they send out, through a series of proteolytic cleavages, sort of a proteolytic cascade, a warning signal, kind of a smoke signal, to recruit more phagocytic cells to come out of the bloodstream and come help kill the pathogen that's there. And then for some pathogens, particularly a subset of gram-negative cells, or as we like to say, monoderm pathogens, there's a set of these proteins that actually can bind to the membrane on the pathogen and punch a hole in it and kill it. And that subset of pathogens are called serum sensitive because they're literally lysed in the serum. They don't require macrophages or anything to be killed. It's just this complex of complement proteins that do it. And it's called the membrane attack complex, or MAC, which I always like. So that's the alternative pathway that recognizes these PAMPs, so things like lipopolysaccharide or lipoteichoic acid, things that are on basically every gram-negative or gram-positive bacteria. There's a classical pathway, which is a very similar idea, but instead of these PAMPs, they actually have the classical pathway relies on antibodies to bind to the pathogen to sort of recruit these components of complement.
It's amazingly sophisticated, isn't it?
It's so
the way it works with each arm of the immune system.
Yeah,
amazing.
It is amazing, and I've been told, I don't know if this is 100% true, but this is why microbiology and immunology went together, because people found that, like in sera way back when, this is one of the first things they found, that they could show like sera alone would kill microbes, and they wanted to understand how that happened.
Yeah, most people don't appreciate the abbreviation of MAC. And I always stress to the students, you'll never forget how complement works if you appreciate what MAC stands for. Membrane attack complex. And if you attack a bacterium's membrane, it's dead.
Game over.
Game over. And that's the marvel of MAC and the beauty of the alternative pathway. The other metaphor I use thinking about the alternative pathway is I often say what the C3 actually does, it's effectively the candy coating on the pathogen like an M&M, and no one can resist an M&M to snap it up.
Exactly.
It's that candy coating. And then MAC comes in and bursts it open just like your teeth do when you bite down on that M&M.
I think that's great. One of my a rotation student in my lab told me that he was taught that it was the sriracha sauce.
Yeah.
So whatever you think
whatever
makes
works
tasty,
for you,
that's what C3 is doing for a macrophage.
Cool.
Okay, so normally, again, like these are in sera, right? You're not supposed to have any bacteria in your sera. So there has been over the years some evidence that there are components of complement that are not necessarily in sera. And people have seen prior to this work that levels of, that there is some evidence, again, that C3 in particular, this thing that makes things tasty, has been around in other places and produced outside sort of the blood. And so the big question this paper asks is there a complement system in the gut? And if so, how does it function to maintain healthy microbiota and eliminate pathogens? How does it distinguish friend from foe? And, well, is it there at all? And this paper is really trying to ask that question. And they start with a series of, actually, most of the experiments in this paper are quite straightforward. It's very thorough. There are many, many experiments, but I'm going to try and hit the highlights. The first thing that they do is just try and measure C3 proteins, and they use sera, where they know it should be there, of age and gender matched wild type germ-free mice, so mice that have no microbes in them at all, no microbiota, and another set of mice which are specific pathogen-free. So essentially you have mice that have no microbes and then another set of mice that have essentially a normal microbiome, but they're missing specific pathogens. And they compare them, and they're looking at sera, again, where they know that there should be, no matter what, whether there are microbes there or not, complement, and also the gut. And what they find is that the SPF mice, these specific pathogen-free mice, so the mice with a microbiome, have higher levels of intestinal C3 than the germ-free animals, while the levels of C3 in the serum of both mice, the germ-free and the SPF mice, are the same. And that's really nicely shown in figure 1B and 1C. So again, they see that if you have a microbiome in your mouse, you have C3. If you don't have your microbiome in your mouse, you don't have C3 in your gut, you still are fine in your blood for all the component, for the C3 component. And then they wanted to know, okay, is it that being colonized with a microbiome is actually triggering production of C3 because the germ-free mice, remember, have none in their gut. And so they took the SPF microbiota, the ones from these pathogen-free but otherwise normal mice, and they put them into the germ-free mice. And they see actually in their fecal samples are really fast. In 2 weeks, they see comparable levels of C3 in the donors as in the recipients. So basically, taking the microbes from a mouse with them, from the microbiota, and putting it into a germ-free mouse induces production of C3 in the formerly germ-free mouse. They then do the opposite experiment. They give antibiotics, broad spectrum, to mice that have a microbiome, and they see the C3 levels drop to the equivalent levels that they would be in a germ-free mouse. And they also don't see any more microbes
I'm
in their
convinced.
feces.
I think this is worth studying.
Oh, yeah. It's very cool.
Me. Yeah.
I was also a little shocked. I didn't realize that these broad-spectrum antibiotics completely clear out your microbiome to the point where your levels of bacteria in your feces are undetectable. That was a little disconcerting.
Well, they probably are giving the poor mouse more antibiotics than you normally get.
Yeah, possibly. In any case, I was a little
disturbed
by that a little bit. Because I always thought it knocked it down, but not completely out. But this is gone so. And what they also found is that if they looked at mice from different vendors, because you buy your mice from Jackson Labs or wherever, when they got mice from one vendor versus another, they could see different C3 levels in those mice. And then when they transplanted them into the germ-free mice, the microbiota from these mice from different vendors, they saw different levels of C3 that correspond to the donor. So if your microbiome has a mid-level of C3 in it and that's transferred to a germ-free mouse, it will have another mid-level of C3. But if your microbiome is associated with high C3 levels, and you transfer that microbiota to a germ-free mouse, then the germ-free mouse will then induce a high level. So it's
definitely tracking with the microbial population.
Exactly. Amazing. Yeah. Amazing. They saw this also in humans. They measured C3 from healthy human donors without chronic gut inflammation because they're trying to, you don't want inflammation, and that will make it much more difficult to tell where the C3 is coming from. So they have healthy donors, and they find variation in human C3 levels. And when they transfer those to germ-free mice, they recapitulate the C3 levels in the original host. So the human to mouse transplant, the mice that get the human microbiota, their C3 levels reflect that of their human donor, which is also incredible because, human and mice are not identical in terms of what grows in the microbiome. When you do a transplant like that, it's not a perfect one-to-one. So it's still very impressive. Okay, so that's all great. Then they really want to know what's the thing in the microbiome that's inducing C3 levels and dictating how high it is. And it's confusing, right? So some mice have higher C3 levels than others, and it's microbiome dependent. So they want to go in there and figure out what it was. So they do this somewhat complex experiment, but they house mice from sort of different sources together, with one that has low C3 and one that has high C3, and they see that the mice with low lead to increases in their baseline production. And then they want to identify.
And can I just add,
yes?
The reason that experiment works is that mice are what we call coprophagic. They eat poop, and they'll eat their own poop. They'll eat somebody else's poop. And so it's an easy way for experimentalists to vary the microbiota by just mixing the mice in the same cage.
Yes, that's actually, I forgot about that. I was wondering how this worked, but that is a very rapid transfer, and it's that the mice are doing their own fecal transfer experiment.
And you don't need IRB approval. They do it naturally.
That's right.
Thank you, mice. Again, thank you, mice. We owe so much to mice. They do something they call triangulation, which I won't detail, but to identify what the cause is And they see it's basically differential abundance of Prevotella species that seem to be most, mostly Prevotella, but they see some other organisms less familiar with, one called Racaniello. They see lactobacillus, which is a pretty common bacteria group, and they're more abundant. So these three are more abundant in the microbiota of mice. The mice that essentially receive the low C3 mice that then become high C3 mice. And then they want to look in humans. They do the same experiment, and there they sort of find, again, Prevotella. And they actually find that the actinomyces are negatively correlated, but Prevotella is positively correlated. So again, Prevotella seems to be the common denominator. And then they isolate some Prevotella, and they show just moving the Prevotella can induce low to high in this mouse with microbiota with low C3. If they give them just the Prevotella, those will increase the amount of C3. If they use a control, which is just another bacteria called Proteus mirabilis, they don't see an increase. And they see similar things when they colonize germ-free mice just with Prevotella. But really just this one bacterial group, the Prevotella, if they take it from if they put the Prevotella in a mouse with a low C3 microbiome, that's enough to induce it, which is
this
really
must have been so exciting because, again, we've thought about complement just being a system in the blood, but here they're finding clear evidence that our gut, too, has a complement system, and it's determined in part by the microbiota. So I bet they're curious to hear how that's working. Who's doing what in the gut?
Yeah, exactly. Who is doing what? But the fact that they could so quickly nail down one bacteria as sort of the major thing in both mice and humans is very impressive. But it says that, some of the things that are fine-tuning this are the microbiome works really well to keep out bad things. And so that it's just these few things is very cool.
And it's interesting that Prevotella is it's a strict anaerobe and it eats sugars for a living, but it's also inflammatory. And
right.
so you wonder if that's the reason it's so good. I know in the oral cavity, if you have Prevotella wandering around your gum line, you'll develop gingivitis and then ultimately fall into the trap of periodontitis. And so the question then becomes, is it's the Prevotella just aggravating the cells that are producing these immune complex molecules that are going to say, okay, this is bad. I don't like inflammation. Let's get rid of it. And that could be the trigger, having too many of the bad actors. And it goes all back to friend or foe.
Right. It's very impressive in that it's so specific to Prevotella because, again, there are a lot of organisms in your gut, and that it can somehow decide, oh, that's the bad one is very interesting. They go on to try and figure out which are the cells that are making C3, because again, normally these are coming from the blood. And so they're wondering what's making it in the intestine. This is actually pretty heroic. They do RNA-seq and some other experiments. And they find that it's stromal cells. So these are the sort of cells that are part of the connective tissue that is in there. And they're the ones, specifically in humans, they identify the mesenchymal stromal cells. So it's a pretty small set of cells within the intestine that produce these. And on the innate side, they basically, they do also a pretty careful look for other components of MAC, because the experiments I told you about mostly were for just C3. They don't see the proteins that are important for MAC, this membrane attack complex. And really, it just seems to be C3 is the most important of these guys.
So if I could interject some of the first authors' thoughts about this. You just talked about the type of cell in the gut expressing C3. In fact, they said that this was a truly exciting moment to discover that stromal cells are the cell in the gut expressing C3. They had thought that epithelial cells were going to make it, and they did a lot of experiments. But even if they knock out C3 in epithelial cells, they still had C3 protein in fecal samples. So they started exploring other cell types. They got C3 reporter mice from a collaborator and did single-cell sequencing, flow cytometry, and imaging, and they found that indeed it was the stromal cells, which are in the lymphoid follicles, the dominant cell population,
incredible.
Expressing C3.
And they just haven't gotten much attention, stromal cells, not from the microbiology and immunology community. So this is neat, new biology.
Yeah, and these follicles, she points out, their development are dependent on the microbiota. They're basically immune hubs that interact with other cells. And she says it makes sense and opens up new avenues for exploring like how these cells interact with dendritic cells or B cells.
Right. So, I mean, this is sort of like these guys that we've kind of ignored. They seem like they might be the conductors, like the things that are sort of making the microbiome symphony and, getting rid of things that aren't good, keeping things that are good, sort of modulating the immune system. So you can have this very diverse and elaborate microbiome, but that keep out the bad stuff.
It was the Cinderella effect. The poor lymphoid follicles are the Cinderella effect. They're the stepsisters that don't get asked to the ball. and then, the major players that we have been investigating for decades, get all the attention. And these poor little stepsisters are really the ones that Prince Charming is using to control our well-being. It's really determines how. Is
Prince Charming in control, or are they doing it themselves? Are they independent actors?
I think they're independent.
I think they're independent actors. No Prince Charming needed. I'm with
liberated.
It's just the truth, Michael.
That's right.
They're independent actors.
That's right.
Okay.
We're having fun.
The microbiome is very fun.
Well, no, I think it takes us back to the wonderful title, is that gut complement induced by the microbiome combats pathogens and spares commensals.
Once again, the microbiome is in charge.
Yes.
Exactly.
Well, we rely on the microbiome for a lot of our digestion and
vitamins,
metabolism.
Right, and then we've, it has mental health implications. Some of the microbiome. Right. I mean, I feel like we don't even understand the half of it. But going back to this amazing paper, they wanted to really know what C3 was doing. Okay, they know it's there. We can speculate all we want, but is it actually modulating the composition of the microbiome? And they first see if C3 actually is important for for pathogens that cause disease in the gut. And they use Citrobacter rodentium to essentially see if making C3 is important for protecting you from a gut pathogen. And Citrobacter, I did not know this, but it is the mouse counterpart to enteropathogenic E. coli, sometimes called EPEC, and its even worse cousin, enterohemorrhagic E. coli, called EHEC. And they infect weaning age C3 deficient mice and age-matched wild-type controls with Citrobacter. And the wild-type mice all survive. And the C3 mice, actually, half of them die. A lot of them lose weight. And so making C3, in general, seems to be quite important for surviving infection with a gut pathogen. It's not that, C3 is important, but so that was the first thing. But now they have C. rodentium, Citrobacter, as a way of looking to see whether mucosal C3, remember, it's like the salt or the candy coating or the sriracha on top of the pathogen. And so they want to know, is the C3 really interacting? Is it just you need C3 generally to fight Citrobacter, or is it in the gut? So they have GFP labeled Citrobacter rodentium. So it's basically a GFP green fluorescent protein in these bacteria. And so they can track them in the gut. They can see them in the gut. And they see that these GFP labeled bacteria within 7 days of infection in a wild type mouse now, they can see that they are coated with C3. So the Citrobacter actually are being coated with C3, which says that the is consistent with C3 is actually making them look tasty, or the word to the macrophages. The word we, teach our undergrads is opsonization.
Mm-hmm.
Yeah, which I think in the Greek means make tasty.
Exactly.
Yes.
I believe it does. In case you end up, needing to use a little ancient Greek somewhere.
Yeah. And I'm sure this experiment struck fear into the hearts of the Iacocca committee, because if the Citrobacter rodentum got out of containment, it could literally wipe out a mouse colony. So I'm sure Iacocca was all over these experimenters to make sure they had good aseptic technique.
Yeah. Well, I was interested, though, because they said that the wild type mice all survived. So it was only the C3 minus that died. They still want to know, though, is it the C3? Where is the C3 coming from? And then they do this super clever experiment, which they use cobra venom factor, which apparently depletes C3 in the plasma, but it leaves the intestinal C3 alone. So if you give cells or mice cobra venom factor, they will no longer have C3 in their sera, which I would guess would be a bad thing. But the intestine, it doesn't have any effect. And they see that when they do the CVF treatment, this cobra venom factor, it doesn't actually compromise resistance to C. rodentium, to the Citrobacter. It's because there's still C3 in the gut. So that says that the C3, right, the knockout mouse, you can't do that, but this is nice and takes it just out of the serum, that it's a C3 in the gut that's helping the mice survive the Citrobacter, which I just thought was super clever.
And it's reassuring that our guts have got that extra layer of defense, right?
Exactly.
Not just a structural barrier, but complement system.
And they show that levels increase during infection. When they infect with Citrobacter, they see the levels increase. And the C3 in the gut, they can also see it stimulates neutrophils to attack. They use that same GFP-labeled C. rodentium. And then they look at the neutrophils, and the neutrophils have basically, when there's C3 around, the neutrophils are better able to engulf these, to phagocytose the C. rodentium. So the complement C3 is working as it should to make the pathogen look more tasty to the phagocytic cells. Yeah, so I just thought this whole thing was really neat. I mean, they take it from seeing the C3 to showing that it's important and varies with microbiome, and that it's, can be induced by pathogens or Prevotella, and that it actually works specifically in the gut to combat gut pathogens.
Yeah. And it's comprehensive. It is really rigorous. It's very impressive piece of work.
So another eureka moment for the two first authors was when they discovered that the genes responsible for encoding the proteins, and this was a few minutes back, were not expressed in gut cells, which are the majority of colonic cells, both in a homeostatic environment and during infection. And despite upregulation of C3, they couldn't find any protein levels in fecal samples. So they concluded the MAC pathway doesn't function in the gut, explaining why C3 had minimal impact on commensals. And she says this puzzled us for a while, especially when we saw that the microbiota in mice without C3 was pretty similar to those with it under the homeostatic environment. So basically, without a working MAC pathway and with not many phagocytes hanging around, C3 might tag commensals, but it probably doesn't actively clear them like it would if MAC formation or phagocytosis were involved.
Cool.
Very cool.
So this is a big collaborative study, they write, between many labs, with the Casper lab, the Chu lab, the Mechalanis and Moffitt labs, all in different departments. Also collaborators from Boston Children's Hospital, University of California, Riverside, Genentech, NIH, even Wash U, and the Cleveland Clinic. And they are two first authors, as you heard. Meng Wu, originally from China, who went to undergraduate at Tsinghua University, got a PhD with Jeff Gordon at Wash U, and then did postdoctoral research in the Casper lab at Harvard Medical School. And she recently joined the department at Wash U as an assistant professor. So now she's your colleague, right, Petra?
She is now my colleague. I met her for the first time
That's cool.
last week, and she is a big TWiM fan.
Yeah, she said she likes baking, running, and listening to podcasts, including TWiM. And then we have also the other, Wen, the second co-first author, originally from China, both PhD and postdoc at Harvard with Dennis Kasper. And some advice. Both have a lot of advice. First, so Meng says, collaboration is essential. It's like the secret sauce that makes everything come together, and you can find collaborators in different ways. You stumble on them, you find a new technology, you bump people into people in the hallway. And she writes, that's where the magic happens, when ideas are shared and challenges are tackled together. It really does take a village to do science.
I 100% agree with that. Yeah. I 100% agree with that. There are no geniuses. There are just collaborations that result in genius ideas and papers.
And then another thing she wants to share is that when it comes to career paths, there's so many options. There's no right way to go about it. So she finished her PhD, decided to take a little detour, worked in industry for a year to see what it was like, and it helped her figure out that her passion lies in academic research, and her PI took her back and gave her a chance to do something different. She says there's no one-size-fits-all when it comes to careers in science. We have to follow our hearts and do what feels right for us. And then Wen has some advice to all the young scientists out there. My advice is never, ever give up. And she shares her journey. She went through at Harvard Medical School PhD. She went through multiple projects. And she says, I'm talking 7th year PhD program struggle vibes here.
Ouch.
I felt like I was constantly wrestling with both my passion and frustration. By the 5th and 6th year, when it felt like everybody else was out there publishing papers with big names, I was still in the lab, feeling like I was swimming upstream. But you know what? Suddenly, everything clicked. Ideas flowing, experiments working. Turns out learning from failure and gaining experience really does pay off. And so her two major projects, the gut complement and microbiota targeted maternal antibodies, worked out. And then she said, I approached her advisor, Dr. Kasper, about continuing as a postdoc, and she was able to do that and collaborate with Meng to do this project. And interesting, after her time at Harvard, she founded NanoCure, a biotech startup based in Hong Kong, focused on antimicrobial peptide drugs and live engineered bacteria for therapy with checkpoint inhibitors in immuno-oncology. They've raised money to do that, and that sounds great. Besides science, she likes to do horticulture, snowboarding, or her other passions, growing plants, another form of science, but with much higher success rates. It helps me balance out the failure rates. She has a large collection of succulents under grow lights.
Good for you. I agree, Wen. And that's why I say we call it research and not search.
Mm-hmm.
A lot of until it works.
I'm going to totally use that.
You can quote me.
Yeah. All right. Thank you, Petra. That'll do it for TWiV. No, this is not TWiV.
No, it's not TWiV, Vincent.
I do TWiV a lot, so I'm kind of used to that.
That's right.
That'll do it for TWiM 305. If you like what we do, we'd love to have your financial support. Microbe TV, the production company of all these podcasts, is a 501(c), which means it's a nonprofit corporation. In the U.S., your contributions are federal tax deductible. So whether you give $1 a month or $100, which some people do, and we're very grateful to you for doing that, it is tax deductible. And we need these donations because we don't do ads or anything like that. We bring you science, the truth, for free. And if you have any questions or comments, please send them to TWiM@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure. Good to see you all.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin's at Washington University in St. Louis. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv, or you can find me here at the Incubator, which is in New York City. If you want to visit, many people visit. Just contact me, and we can set up a time for you to see this cool space where we build spaces for appreciating science. I'd like to thank the American Society for Microbiology for their support of TWiM and Ron Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.